Okay, you're welcome back. So let's look at Gregor Mendel, the man that started all this experiment. Like I told you, the man doesn't have any information about um, DNA, so that's why his work wasn't um, noticed until 34 years after his death. Some other scientists came in and started researching and started discovering more things from what he had. So that's why they gave him the credit. They are calling him the father of genetics. Okay, this man is a farmer. Let me put it that way, because from the story, he owns a farm and he used to plant one particular crop seed called peas. Now, one of the days he discovered that some some pea plants came out to be short, while some are tall. He also discovered that the seed they produce, some are spherical or some are round, while others are wrinkled. He also discovered that some are yellow and some are green. He also discovered that the flowers are some are purple, some are white, and so on and so forth. He discovered so many things, and that's what created the curiosity in him that made him start asking questions, start researching to know why. And one of the things he did was to he identified two pea plant, one tall one and one short one. Okay. Now, when he identified the two, he now said, okay, let me cross them, let me crossbreed them. So let's assume that this is the plant one and this is the plant two that he identified. Okay. Now he collected the pollen grain from this plant one and transferred it to the stigma of plant two. That is the process of pollination. Now, when he did that, the pea plant produced seed. Now he now collected the seed and then planted it again in the soil. Now, when the plant grew, he discovered that the plant grew to be tall. So he was now wondering, so what happened to the short one, the dwarf one? Why is dwarf one not showing? Why is the plant tall? Okay. He didn't stop there. He continued. He collected another sample and but while as he's doing that, he's coming up with a conclusion. Now he said there's something in that top plant that's overshadowing the one in the dwarf plant. So that's why he came up with this big T, small t. So he assumed that this top plant is a big T, or this can be big T, or big T, small t. But it can't be big T, small t, because if it is, chances of the next plant coming out short will be there. So this is small t, small t. So all is resolved. By the time you cross this and this, it's going to give you big T, small t. So all the plants, the new plants he produced, all of them came out to be tall. Okay. Now he continued. He picked two again from the sample. Remember I told you all the F1 here is going to be heterozygous tall. So he now picked two heterozygous tall from and crossed them again. When he crossed them, he discovered that one of them came out to be short. So he now said, yes, this is because these plants are carriers of short gene, but it's not manifesting because of the dominant gene. So that's why dominant allele, sorry. That's why he came up with the idea with his first law, which he called law of dominance. Now, law of dominance is when the dominant alleles overshadows the recessive alleles. Okay, so in the presence of a dominant allele, there is always a masking of the recessive alleles. Okay, so that's why the plant happened to be tall, despite the fact that it's carrying a short gene. So this is the first law. This one is called law of dominance. Now let's see more about this law. He said the heterozygous individual has different alleles. The one that appears in the offspring is dominant allele, while the one that appears in the one that does not appear in the offspring is called recessive allele. Excuse me. Okay, so example, you mentioned some of the dominant alleles. For example, we have brown eye. Brown eye is mostly dominant over blue eye. So anybody you see today with blue eye, that means the person is carrying small b, two recessive alleles. Anybody you see with big eye, with brown eye can be this or can be this. Okay, so in this case, dominant will overshadow, while in this case, both of them are dominant. Okay, same thing with rolling of tongue. Some can roll tongue, some cannot roll tongue. Me, I cannot roll to my tongue, so I don't know about you. Now, I also have brown hair. I have the one that's blonde or red. There's some can 6 fingers, 5 fingers. So 6 fingers is usually dominant, but it's very rare. It's very rare. Okay, so anytime the gene for 6 fingers shows, it always manifests. It always manifests. Okay, so that's the first law. Now the second law is what we call the law of segregation. Now this law of segregation is the English word segregation means to split. The English word means to split. Remember, male is we are carrying XY chromosome, while female are carrying XX. Now during reproduction or during meiotic stage, now if you don't know about cell division, you can refer to some of my videos on cell division and study about them. Now cell division is of two stages. We have what called the mitotic stage, and we have what called the meiosis stage. Now meiosis stage is one that deals with reproduction, while mitotic stage deals with body growth and development. Okay, now for meiosis there is a splitting. Now when this man and this woman wants to make babies, now this man is going to split what he has to produce X and Y chromosome, while the woman is going to split hers as well to produce X and X. Okay, so the man's X or Y will approach the woman's X and then fertilizes to produce the baby. So we call that recombination. Now recombination is always a process within the meiosis. Like I said, I don't want to go into the topic meiosis. You can refer to the previous videos to know more about meiosis and mitosis. Okay, so the law of segregation is when the alleles separate. So this one is also referred to as monohybrid. Remember, we're dealing with one trait now. And what's the trait? The trait here is sex. So the sex can be either male or female. Okay, all right, so I think that's for law of segregation. Then the next one, which is called the law of independent assortment. Now this one is a little bit broad, but it's still very simple to understand. When we talk about independent assortment, we said that these things are sorted independently. There is no laid down rule. Okay, for instance, okay, this one is also called the hybrid, because here we're dealing with more than one trait. There is usually two. Okay, someone will say, is there anything like tri-hybrid? Well, I'm not aware of that. I don't know. But if there is something like that, good. So this is dihybrid cross. Okay. Now, for result of this experiment, Mendel derived the law of independent assortment. Now, the law of independent assortment states that allele pairs separate independently during the formation of gametes. So they separate independently formation of gametes. This means that traits are transmitted to offspring independently. That's why you cannot know which one, if you're going to have a male child now or female child. I believe that's one of the reasons. So it's done independently. There is nothing controls how it's been which one, because during reproduction is the one that gets to the egg, fertilizes the egg. So everything that happens is done independently. They are not depending on any factor. Okay, so that's law of independent assortment. Let's see an example here. Now if you look at this, remember I made mention of this when we did the hybrid cross. Now I mentioned the two traits, which is the color of the seed and the shape of the seed. Okay, so big Y stands for yellow, while the big R stands for round. The small y stands for green, while the small r stands for wrinkled. Okay, now if you look at this, let's look at this. Now these four combinations are the possible combination you can get from this. These four combinations are the possible combination you can get. I just want to tell you how they drive this table. Now this is how I do it. Now if you don't understand this, please still make use of your Punnett square and your cross method. You can use that to get your answer. Okay, now that it's like this, what I would normally do, I will draw my line. I will draw a line like this, and I will call this, I will identify this big R, big Y, big R. That is this one. Then I'll do another line to this place, so I'll call this big Y, small r. So that is, am I correct? Yes, that is this. Okay, now we'll go again. We'll go on the next one. This is Y. You cross this like this. So this one I have small y, big R. Do you have it here? Yes. So this is it. Then you go to the last one, you cross it again. This is small y, small r, and that is this. So these are the four combinations. These are the four combinations you can get from this round yellow round seed. Okay, now this is the Punnett square. but Punnett square can go more than 4 cells. Remember, before we use 4 cells because we are dealing with individual monohybrid. Okay, but here now this is dihybrid, so you do this dihybrid with more than 4 cells. So here we have 16 cells. 16 cells. Now if you look at this side, you will see the result. Now they've crossed it. Now how do they go about crossing it? After arranging, you can see this will go with this. Now if you look at the way they arrange it here, you can leave it this way, or you can still do it in another format. You can do it big Y, sorry, big Y, big R, big Y, big R. So this, they're all the same. This is the same with this here. The only thing is, this, for the sake of interpreting, that's why you write it this way. But for me, I will know what to interpret here, because this is yellow round. 2 dominant yellow, 2 dominant round, finish. Okay, so the same thing goes for next one. You can see do it this way: big Y, big R, small y, big R. So this is still yellow round because of the law of dominance. So you can practice the remaining ones and see how good you are with it. Okay, now look at this number again. Look at this number 9. This number 9 here. Let me show you how God came about this number. Now this is showing you big Y, big R. So this shows yellow round seed. So you go into the cell and pick out all the ones that is carrying yellow round. Now this image here showed you use color description and imagery. Most times they might not give you color description. They expect you to find it out by yourself, by just giving the genotype. Okay, so what you do, you come here, you start crossing them out. Find all the ones with big Y, big R. So this is one. I'll cross it out. This is another one. This is, I'm counting 1, 2, 3. Sorry. This is 3, 4, 5, 6, 7, 8, 9. So that's how I've got 9. So the next one is yellow. Yellow is small r. Sorry, yes, green. Sorry, no. Green is small y, and is smooth. So that means this is round. So this is green round. So we identify green round again. This is, you count 1. Don't count the ones you've counted before. That's why you're crossing it out. This is 2. And this is 3. So that's why I have 3. So next one is yellow wrinkled. So you find the one that's yellow wrinkled. This is 1. This is 2. And this is 3. Then the next one is green wrinkled. So this is just this. So that's how they came about this number you have here. Okay, so you can try this out. You can try this out to see how far you can go with it. All right, so next video we'll talk about the pedigree and we'll look at some questions in genetics. All right, so see you in the next video. Bye.